thank the organizers to give me the chance to present my work, which combines a disease analysis and protein-protein interaction data. Parkinson's disease is a complex disease that most likely affects someone that you know. It's very complex, and in the last years, a large number of underlying dysfunctions have been identified. Nevertheless, it's still unknown what actually triggers the disease or what type of disease subtypes exist. What is known is that Parkinson's most dominantly affects the synapse, which is the structure you can see on the slide on the right. The synapse is a structure that is divided into two main regions, the pre and the post synapse. So we present it with a very complex system and a very complex disease. And we've decided to use protein-protein interaction data and analysis to shed light over the complexity. And by doing so, we would like to answer two questions. The first is to find evidence that Parkinson's disease actually significantly affects this region. And if that is the case, it's also very likely that the disease affects a certain part of the structure, certain synaptic regions. And if that again is the case, we were wondering what the general function of those regions is. In the next couple of minutes, I'm going to tell you about the data that we've been using to carry out this analysis. And we've very largely stressed the quality of that data, because data quality is crucial to then draw conclusions specifically on protein-protein interaction networks. So I've mentioned Parkinson's is a complex disease, and some of the underlying dysfunctions are mitochondrial dysfunctions. The ubiquitin system is largely affected, and the vesicle cycle as well. We've used data from a number of databases, and we're able to define a key gene set of about 418 genes that have genetic or curated evidence to be linked to Parkinson's. So the next data set that we needed was the protein data that describes the synapse. We've chosen to use proteomic data because that actually is the data that tells us which proteins are expressed in that region. And in a joint effort in the lab of Douglas Armstrong in Edinburgh, we were able to review most recent proteomic synaptic data sets, and we defined 3 proteomic regional synaptic sets, where one of them defines the pre-synapse, the second one the post-synapse. We've got a synaptosome, and we also joined those 3 data sets to have a general overall synaptic data set. You see the numbers, they range between about 1,800 up to 6,500 proteins. They might be quite large. I'm very happy to have a chat with you later why this is the case. And amongst those, we could see that there's 205 disease-associated genes. So this helped us to answer the first question. And we've used hypergeometric testing to significantly show that finding 200 proteins that are associated to a disease in the set of synaptic genes based on a genetic background is significant. So this then allowed us to tackle the next question, which was basically linked to building protein-protein interaction networks. And therefore we need protein-protein interaction data. Again, following the principle of highest data quality, we used information from 4 different databases. And after mapping interactions to the same identifier, we were then able to filter it for only human interactions, interactions that were detected based on a direct interaction. We excluded spoke-expanded interactions, and all of the interactions we have a PubMed ID as a reference. Based on those, we were then able to generate those 4 synaptic protein-protein interaction sets or networks. They also, again, range between 1,500 to 6,000 proteins, and we're talking about somewhere between 10 and 70,000 edges. So these are our networks that we want to analyze, and how can we divide them to find the substructures where Parkinson's is mostly affecting this region? So we used different clustering algorithms, and the 5 of them that we had chosen are FastGreedy, InfoMap, Louvain, Spectral, and SpinGlass. So they all based on different approaches, but what they all do is that they split the networks into so-called communities. So based on the 4 networks and the 5 algorithms, we came up with 20 different network topologies and a large number of communities. And we asked the question, which of these communities are specifically affected by Parkinson's disease? So again, hypergeometric testing helped us, and we analyzed every individual community. We counted the number of disease-associated genes in those communities, and we compared that with the number of communities and the size of the respective network. And this showed us that there's 41 communities that are significantly disease-enriched. One might ask, are these communities in networks that have been generated or divided by the same clustering algorithms? One can also ask, do they come from specific data sets? There are slight biases, but there's no obvious trend. So we decided to compare the proteins in those disease-enriched communities amongst each other. And we started with the Parkinson's disease-associated genes. So we found 83 of them. You can see them at the bottom of the heat map bar plot. And every row represents one community. So what we then did, we clustered these communities, and we can see that similar communities cluster together. I've highlighted the first one. There's a second one here, and we can see a third one there. So this basically tells us that there's a large overlap between disease-associated genes that have been clustered together based on different data sets and based on different clustering algorithms. But the disease genes always appear in the same network communities, meaning that they're most likely interacting and showing a high effect. To go one step further, we also considered all the proteins in these enriched communities. And again, you can see the proteins on the x-axis, and we could identify 3 clusters. We then checked which of the communities in these, which are the communities in these clusters. And it was nice to see that those are exactly the same communities that we see when only comparing the disease-associated genes. We had a closer look at those clusters. Since they made of several communities, we considered the coverage of the proteins in these clusters. And our first cluster shows 90 proteins that appear in at least 2 communities, out of which 6 of them are associated to Parkinson's. The second cluster is about half the size, with 5 disease-associated proteins, and the third cluster is relatively small, but we can still see the combination of those 2 disease genes over several communities. We then had a quick look at what these Parkinson's disease proteins actually are. I'm not going to go into any detail, but these are not the most suspected Parkinson's disease genes that one can think of. So we then went on and asked, what is the overall function of those communities? And we used gene ontology enrichment analysis. We combined the Fisher exact test with the Alexa elimination algorithm, and we also corrected our results for multiple testing. And what we can then interestingly find is that community cluster 1, is highly enriched for neurotransmitter secretion, scaffolding protein binding, and the pre- and post-synaptic membrane. Our second cluster is enriched in immune response proteins. And the third cluster shows neuromolecular junction development and calcium channels as being highly enriched. What does that tell us? We've identified 3 different synaptic regions with a number of proteins, and some of them are associated to a disease. Going one step further, they then also tell us that those clusters are actually in charge of different function in this complex system. So I would like to summarize by saying that data and data quality is very important. And through the use of protein-protein interaction data, disease data, and the proteomic data set, we were able to build these networks and cluster them. Using different clustering algorithms helped to then be able to be more specific and more certain about what we can find is actually true, or very likely what is happening under biological conditions. Analyzing these findings and using functional information helped us to generate the new disease gene set, which we could also interpret from a functional point of view. This should now help us to carry out more targeted future Parkinson's disease analysis and identify disease subtypes, disease triggers, disease biomarkers, and drug targets. And due to the open system that we've been implementing, this is also just a framework to ask other complex questions, to use other data sets and disease data sets, and to hopefully help advance the research in complex diseases. Said that, I would like to thank my group in Edinburgh, and specifically Oksana and Colin, my research group in Stockholm, and my supervisors there. Funding is coming from a Eurospin Erasmus Mundus program, and I'm very happy to take any questions and have further discussions over my poster later today.